Now, because Edmond told us that pathology is impossible for computer to decipher, I changed the name of the talk, and it's not computational pathology, but rather computational oncology, although we will focus on pathology. Okay, so, and believe me, oncology is probably as complicated, or at least the same level of complexity as pathology. So what can we do? First of all, a little bit of background about myself. I am still doing geometry analysis and things that have geometry in them. And I will comment later on in this talk about what is geometry and what do I mean by that. But let me just summarize what Edmond was talking about, at least how it is projected on my engineering or mathematician brain, trying to simplify everything to a really, really simple process. So usually when somebody feels bad or suspects something, he or she goes to a physician, to the hospital. If the physician suspects something, then he or she would take, the clinician would take a sample. Usually the first thing would be to use an ultrasound in order to guide some injection that would pull up something which is called needle sample, because the sample is ejected through a needle. And then the simplest and most trivial way of looking at this sample would be to put it on a slide and color it, stain it with something which is called hematoxylin eosin. And this is the beginning of the detective work, the complicated and tedious and intellectually demanding work of a pathologist. So you take this piece of tissue, really thin slice, you put it under a microscope and you look at it with your two eyes. And then you scan through resolution and you use your whole knowledge of a medical doctor to understand what is going on inside. You start from the structure, from the architecture, then you dive into how the cells and the surrounding of the cells are interacting with one another. And in order to become a good pathologist, you need to train a lot. And unfortunately, pathologists are a rare commodity. I mean, the population grows in a much more rapid way than the number of pathologists that you could find, okay? And the question is, can we somehow bridge this gap? And I would not say that we can. I would not say that we could, not in my lifetime and not in Edmond's lifetime. But what I would try to convey is that there is a methodology, there is a way that we could think of helping pathologists along the way. Now, to calm Edmond down, I would talk about deep neural network, about how this kind of new emerging field in computer science can be used in order to help this way. And the FDA, the Federal and Drug Administration in the US, would not give a permission to a system which cannot explain its decision. So as a supportive system, as something that pathologists at the end of the day would sign on what is going on, we could probably do something. But as a standalone replacement for a pathologist, this is far and beyond our lifetime. At least this is what I think. So once you have the hematoxylin and eosin slide, the pathologist is looking at the slide and then starts the detective work. The pathologist decides whether there is cancer or there is not cancer, but then he or she has to decide what is the kind of receptors that you would see in this cancer. So if we are talking about breast cancer, and we have here Karen, who is an oncologist expert in breast cancer, you have to decide whether there is estrogen receptors, progesterone receptors, et cetera, et cetera. In order to design the treatment, hormonal therapy, biological therapy, these kind of PD-L1 blockers or PD-L1 inhibitors that you would have to apply to the patients. And along the investigation, you would stain the tissue, another slice of the tissue, with more complicated kind of stains, immunohistochemistry or next-generation sequencing, I mean deciphering the DNA. And at the end of the day, you give all this information back to the oncologist that would decide on the proper treatment. Should it be chemotherapy, biological treatments, just cutting out the tumor out, radiation, et cetera, et cetera. And the question is, can we help pathologists and oncologists along the way? Not replacing them, not deciding for them, but just helping them a little bit and accelerating the work. Just a word about our team. Dr. Gil Shammai is running the team that is doing all this effort in computational oncology and pathology. And we have a huge group of data scientists and people that are located in some of the hospitals and getting the data. Some of our veterans are now pursuing other careers in Amazon, in Verily, and Omer Wasserman decided to be a pilot. So if you fly El Al and you hear Omer Wasserman, know that he knows a little bit about computational pathology as well. Yeah. We also connected with a lot of clinician collaborators, including Edmond's lab in Carmel Hospital, who have been really helpful in our pursuit. So if you think about deep learning, and in a moment I will describe what deep learning is, think about it as a car that needs fuel, and the fuel is data. If you don't have data, you have nothing to do in this domain. And Edmond and others have been really helpful in getting this data. And if you go to a hospital anywhere in the world, in Europe, in the US, and you will ask for data, you will see how tedious and problematic this process can be, even if you're located inside an hospital. But right now, we have a system that can extract data and can get data. And it's not only the data that matters, it's also the quality of the data. You need the annotation, you need the physicians to define exactly what is going on in the tissue that you're looking at, otherwise your system is doomed to fail. Okay, so what am I talking about at the end of the day? What is our mission? Our mission is to leverage histopathology, pathology of looking at tissues, in order to improve oncology, and at the end of the day, provide better personalized care. I'm looking at yours or hers tissue, and according to what I'm seeing, I would be able to decide what would be a better treatment by saving the pathologist some of the investigation work that he or she should be doing. Okay, so a little bit of background. What are our tools? If you take an image, this is introduction to computer vision, image processing, etc. If you take an image and the only thing that you do is improve the image a little bit, this field is known as image processing. If you take an image and you are trying to understand what is the geometry, the hidden geometric patterns in an image, this field is called computer vision, okay? And if you are trying to look at the geometry, at the structure, the geometric structures of something that you have constructed, and you are trying to create a nice-looking image, this is the inverse problem, and it's called computer graphics. Along the way, there is also geometry processing, which is taking geometry and improving it a little bit. And there is also augmented reality and virtual reality, which is a mix between all these fields that I've shown you here. But this is more or less the issue. And what I've been working in is basically, you don't see the cursor, is basically computer vision. And when I came to the Technion many, many years ago, now it's many, many years ago, they told me that the most complicated problem is the segmentation problem. So you're looking at an image, and you're trying to segment the fence, the lighthouse, these houses, and you're trying to understand what is going on. They told me that if you solve this problem, this is the holy grail in this field, so you have solved everything. And since I was an engineer, I thought you need to bypass this problem. You cannot really tackle it heads on. And the first thing that we did at the Technion, my lab and I were building a machine that extracted the geometry by having something like two eyes. When you have one eye, you just have a projection of the world. If you have two eyes, you have two projections of the world, and the brain is basically finding the correspondence between features in one image to the features in the other one, and the brain extracts the geometry of the world. This is why when we pour water from a pot into a glass, we don't miss. Okay, try to do it with one eye shut, and you will miss. So we built a system like that. It had two eyes, and to make a long story short, we sold it to Intel. This was a real sense in Intel. Intel made it very efficient, very small, very tiny, also cost-effective. It optimized, and this is a comparison of the competitor at the time. In fact, nowadays, you can find these kind of 3D sensors on your telephones. If you have an iPhone, then you have a sensor like that. You have two eyes here on the telephone. And this is how, for example, you can use face recognition in order to open your phone. Okay, so one of the leading engineers and thinkers in this project was, in fact, two of my students, Alex and Michael Bronstein, sitting here in the audience. He will talk this afternoon. So this is one way of bypassing the problem. Would it help us in pathology? I'm not so sure yet. 10 years after, there was a revolution, in fact, a change of paradigm in computer science as a whole, and this was the reintroduction of neural networks. So here I have Jan LeCun, George Hinton, and Joshua Bengio, who got the Turing Award for the work for the reintroduction of neural networks. And the idea is that rather than developing an algorithm per problem, what you'd have is an algorithm, a fixed algorithm, but a very complicated one. And the only thing that you have to do is tune up the parameters of this algorithm to solve a problem. Now, what is this problem? The problem is that you see a lot of data. Data, you have X, and the output would be Y. So you feed this function a lot of X's, and you would train it to produce the corresponding Y's. You can think of X as an indication of the features that you see as a patient is entering the physician, and as Y, the prediction, the prognosis of the physician of this specific patient. And the whole methodology of training the network, of tuning up these parameters, is called training. It's the professional word for that is called backpropagation. You propagate back the gradients of the discrepancies between the predicted Y's and the actual Y's. And if you have your Y's, this is called supervised learning. And if you don't have these Y's, you have to think a little bit of how to do that. And this is what we have been doing in my lab for, I would say, the last decade. So let me just give you an example. After building this 3D capturing device, this sensor that can capture geometry, the question is, can you use a single sensor, just one eye, in order to extract the geometry of a face that you're looking at? So the answer is yes. And this is a result of Elad Richardson. What you see here is a neural network, straightforward neural network. It's called convolutional neural network. Looking at a single image and extracting, predicting somehow something that looks like the 3D reconstruction of the image. And the idea is that if you train the network hard enough with many faces and corresponding geometries, a network would be able to do that. So this is, in a sense, it is an hallucination. It's exactly the opposite way of thinking axiomatically. You don't have an axiom according to which you extracted the geometry, but you train the network with a lot of data. And you can see that, and what you see here on the right is the texture that you're looking at back projected onto the geometry, and then the geometry is rotating in space. And the question is, can we use the same idea of a network and apply it to problem in pathology? And the answer is yes, this is what we have done. So let me just tell you why it is important, even just as a second read. Okay, you have the pathologist, he's looking at the slides, and we use our system in order to verify that we can do something useful. And this is actually, I would not say from which hospital, we actually can recognize mistakes that have been made. So what happened is that we are using our system in some hospitals, including in Edmond's lab. And the system is looking at cases that were analyzed by a pathologist. And whenever there is discrepancy between the pathologist's decision and the system's decision, it raises a flag. It means that now, rather than the fellow doing the analysis, it goes to Edmond, and he is looking at it, and he has more experience, and he should say whether there was an error or not. And as Edmond showed us, even if we just look at the central part, at the analysis part of the errors, we can see in this case that two women were assigned a wrong treatment, and a system like that could actually benefit, even if just deployed today to these hospitals. Okay, so when we stain a tissue with hematoxylin and eosin, it is routinely done, it is very cheap, a couple of dollars per slide, and a physician, I mean, the oncologist, the pathologist can say whether there is cancer or not, what is the type of cancer, and what is the grade of the cancer, I mean, how severe it is. But it is limited. I mean, what they cannot do is predict the genetic mutation, what are the characteristics of the tissue, what are the molecular receptors, and since he or she doesn't know what are the molecular receptors, they would not know what treatment to assign to this patient. So from just the age, and then starts this journey of exploring what is going on. And usually it involves immunohistochemistry at first, and in this case on the left you see similar tissue which has estrogen receptor which is called ER positive, and on your right you can see estrogen negative, and it has to do with how the nuclei of the cells are being sensitive to a specific kind of this kind of material that was extracted by tedious and hard work of injecting various chemicals to poor mice. And I'm not in a journey to save the mice, but I think that we could look at this hematoxylin and eosin and at the end of the day predict what would be the ER positive and ER negative even without immunohistochemistry. So when we feed this kind of decision making to a neural network and we ask it to make a decision, the first question that would come in mind, what are the features that such a system would produce? So again, giving H&E staining slides to a pathologist, he or she would not be able to say whether there is ER positive and ER negative. And the question is, can a deep neural network do this job? At the beginning, when we started that, we had a lot of comments saying, look, the data is not there. You cannot see that. It would not work. And then we trained the system completely legitimately, and at the end of the day, we have shown that statistically you can do it. There was a paper 3 years ago that was produced in JAMA Open that proved that you can do it. And the features that the system was looking at were these regions where we color it artificially with green and red regions, green for positive and red for negatives. And believe me, I'm not a pathologist and I don't know what is going on there, but there are these regions according to which the system has made the decision. So the first work was joint with 3 physicians and 2 of my students, Ron Schlossberg and Gil Shammai. Yoav Binnenbaum is now in the US, Iri Duek and Ziv Gil, who was at the time head of a department at Rambam Hospital. And we have shown that you can actually decipher some of the hormonal receptors from these kind of tissues. It made us, at least locally, famous for a while. But what is important to say is that we have shown that there is correlation, significant correlation, between the H&E images and all the receptors, all the biomarkers that you see here on the left. Okay, so there was a significant correlation. So there is hope of adding more data and training the system. And what you see here on the right is the fact that the more slides we add per patient, the better the accuracy of the whole process would be, which is important. If you think about it, you get a part of the tissue, and usually you slice it at several locations, and these several locations are being examined by the pathologist. You can make a mistake. I mean, you can take a wrong slide, as Edmond mentioned, just at the top or just at the bottom. And then when you color it with immunohistochemistry, you would completely lose the prognosis, the correct prognosis. So the more slides we add, just H&E slides, the better the accuracy would be. This is something that we are really proud of. In fact, some of the new computer scientists are famous by the fact that they had created databases for computer vision to work on. There are famous researchers in my field whose main contribution was constructing a huge database of annotated objects in real world. Right now, when we compare ourselves to the rest of the players in this field, and some of them are quite, I mean, usually there would not be universities, but rather companies. Why? Because what happened? Okay, so the history goes like that. In 2017, Philips pushed the idea of digital pathology really, really hard to the FDA. So until then, it was not allowed for a machine, for a camera to take pictures of the slide. The belief was that in order to understand what you're looking at, you need to look at it through a microscope. You need to play with a focal point, etc., etc. So it was 5 years ago when Philips said, look, enough is enough. The cameras today are good enough. Let us extract those images digitally. And this when the digital pathology revolution started. Revolution in a sense, there are many hospitals where you don't have this process going on, and many hospitals where you do. For example, in Israel, in Sheba, I think it is all being done digitally, and in Carmel, not yet. But we will get there. So 5 years ago, also started the boom of many startups who thought, okay, since I have digital data, let's shift the paradigm and also developed software tools to analyze it. And this is where, when Page AI and Path AI, these two companies that I mentioned here, started. They started in a university and they migrated into being a company. In fact, they raised quite a lot of money. And they have many slides that they reported about. Right now, I think that we have more slides in each and every domain, and we can actually start competing, at least in the size of the data with all of them. Okay, so what should we expect at the end of the day? What we would like to see, what we would like to prove is that we can classify the subtype of the cancer. What would be the biomarker that would have been active? Would I have painted or stained the tissue with immunohistochemistry or doing DNA profiling? Then I would be able to just look at the H&E and do disease prognosis. I should be able to predict the survival time. I should be able to predict the relapse, the risk group, etc., etc. And at the end of the day, the bottom line would be better treatment for the patients. Okay, what we do is incorporate histopathology with current clinical pathology. This is more or less saying the same thing. Let me jump ahead and say that until now we have been the first to predict expression of ER, PR, HER2, etc., and most importantly, PD-L1. PD-L1 is very important if you'd like to assign your patient all these biological treatments. In skin cancer, for example, it would be Keytruda and Avelumab and all these PD-L1 blockers. I'm quite sure that in breast cancer you also have this kind of therapy that you would assign to. So knowing if there is PD-L1 in the cancer, I would estimate, I'm not so sure that there are proof of that, would be a good indicator whether to assign this kind of drugs. Oncotype is a really interesting type of analysis which is done computationally, and I would say it revolutionized a little bit the way women are being or men are being treated for breast cancer. And what you do is you take a piece of the tissue and a piece of the background tissue, you send it to this company, they do DNA profiling, so they extract 21 parts of the DNA, and they would give you a score by which you should take or not take chemotherapy. So you were diagnosed with a cancer, with breast cancer, and now the oncologist does not know what to do. But if you go through this oncotype idea, they would give you a range below which you should definitely take and above which you should definitely avoid. And now the question is, can we narrow down the gap in the middle, okay? The vague range where you don't know whether you should take chemotherapy or not. Recently, we have some recent results about leukemia and PD-L1 prediction. I'm quite sure that it would be eventually published in one of the good journals. I don't know, probably Nature Communications or something like that. regarding expression of PD-L1. So we have quite promising results in this domain as well. I would really like to tell you a little bit more about what is the difference between classical deep learning and geometric deep learning, which is completely different from my story until now. But before I do that, let me just tell you that we have a system which is being tested in 2 hospitals, in Edmond's lab, as well as in High-Mech Hospital. And we were able to completely predict the estrogen receptors and the progesterone receptors from just H&E images. I would say for significant part of the patients with 100% accuracy. Okay, so we were confident for a specific, and we know exactly for which patients we are certain and for which patient we have doubts, which is a really significant tools for physicians to use. We are not a company. I mean, this is a laboratory at the university. But still, this is whom we are competing with. So there are many companies who are dealing with something like that. So Tempus and Roche, these are huge companies right now, and they do DNA profiling, etc. But if you look at the rest of the companies, Page AI, Protea, Path AI, Ibex, etc., they raised quite a lot of funds compared to our relatively large academic funds, and they are pushing forward, trying to do things that we have been pushing for a while now. And what is interesting is that after we claimed that we can do one of the biomarkers, immediately you could see in the page, in the websites of all these companies that they are claiming to do similar things. But I'm happy to say that probably they did before that, but they didn't publish. Relatively to Israel, we are well funded. I mean, we have a lot of agencies that are supporting this research. And this is more or less the story about, I promised that I would finish 10 minutes earlier so that you will have enough time to ask questions as well as drink coffee. But I would like to tell you just a little bit about my other side, and this is geometry analysis. So let me go to Cartesian coordinates, if you remember René Descartes. René Descartes told us, thought of the idea of translating geometric problems into algebra. And how does it connect to the story that I've told you until now? When I train a network like that, it is so complicated that understanding what the network is doing is something, understanding in simple equations is something which is relatively complicated. And the question is, can we try to figure out what these kind of networks are doing? And I specifically chose a very simple example of a network that is trying to classify objects, geometric objects. So assume that somebody is giving me the points that are describing the outer surface of this object and the outer surface of this object. Okay, so you have many points in 3D. And these set of points you feed into a network, and it should tell you whether this is a mouse or a microphone or a table or a chair. It is true that according to the scale you can solve it like that, but assuming that somebody is playing a dirty trick on you and allows you to scale everything to whatever you want. And the question is, how would you do it? So classically, if we go 100 years ago, you would think that, what people have been trying to do is look at the set of points and try to measure something which is called moments of the shape. Okay, so you look at the coordinates, at the x, y, z coordinates. Can you see my mouse over there? No. Yeah, you can. So you would think about the set of x, y, z coordinates, and when you sum all the x's, all the y's, all the z's, you have the center of mass. When you sum them after you square them, you have the second order moments. When you sum them after you have another power, you have the third order moments, etc., etc. And according to these moments, you can find what is the center of mass, what is the main axis. This is the second order moments of this plane here, etc., etc. And when you add them, in fact, you can reconstruct back the shape. And early ways of classifying shapes was to look at these small set of numbers and trying to class them, to put them into, to compare between them and somehow put them into different places. I mean, according to the distances between the moments, you could classify those shapes. Okay, so this is what is called classical or axiomatic way of classifying between objects. A couple of years ago, there was a group at Stanford, Su, a student of Leonidas Gibas, who said the following. If you look at these kind of objects, the most simple feature that you're looking at is a point, XYZ point. Put this XYZ point into a neural network, lift these three numbers into a 1,000-dimensional vector. So this is called lifting. So you take three numbers and you lift them into a 1,000-dimensional vector. So each X, Y, and Z are being lifted into this long vector of 1,000 numbers. And then what you do is max pooling. So you go along each and every axis, and you find the maximal number. And at the end of the day, you get a vector of 1,000 numbers. Okay? And this vector of 1,000 numbers is what the network was using in order to classify the type of shape it was looking at. And I was looking at it as a computer scientist, I mean, and I was saying, this is something crazy. I mean, why do you need to lift and project? This is something which is completely non-intuitive. Can we understand what this network is doing? And I will not tell you that we can understand what the network is doing, but we can attempt to understand. If you take the X, Y, and Z coordinates, okay, before that, a network is really, really good at classifying things. It is really, really bad in multiplying two numbers. If you try to train a network to take X and Y and multiply X by Y, or A and B, okay, don't want to confuse with the X from the previous story. So you get a number A and B, and you would like to train a network to find the multiplication of A and B, the number of layers, the number of complexity of the algorithm would be as complicated as the number of bits it takes you to represent each and every number. So you need really, really, really long networks in order to multiply two numbers. If you bear this in mind, then this lifting, this network that is taking each and every point and lifting it into a 1,000-dimensional space would intuitively take the polynomial expression of this X, Y, and Z coordinates. So it would basically give the ingredients for computing the first moment, the average, the second order moments, etc., etc. So assuming that this is what the network is doing, at the end of the day, max pooling is more or less like averaging in a sense. So in a sense, it is basically computing moments. With this understanding of the analogy between moments and this something which is called point net, we thought, can we accelerate, can we help the network and make it more efficient? And this is indeed what a student called Joseph Rivlin, Joseph Rivlin did. So what she did is the following. Rather than inputting the X, Y, and Z coordinates, she also changed the network a little bit and entered not just the X, Y, and Z, but also the powers, the first ingredients of computing the moments for this network. And the network was doing exactly the same. So it was lifting each and every 9 numbers rather than 3 now into a 1,000-dimensional space, max pooling it, and then using that for classification. The end of the story was that we got much, much better result with a much, much more efficient network for doing the same tasks. So she got really good results. It is something that we did about 3 years ago, but she got really nice-looking results in classification rates, and the efficiency of the network was much, much, much higher. Some people would call it handcrafted features or manipulating the whole purity of a network. But this is something that you would do anyway. I mean, when you look at this surface, usually you feed it with normals or local neighborhoods. So you do these kind of things anyway. And I think that this is a way of trying to understand what a network is doing and helping this sort of brain, sort of black box, doing it a little bit better. We have seen that this idea is so efficient, so another student of mine, Oshri Halimi, took 2 shapes. This is yet another way of understanding what is going on. So this is really complicated slide, so let me jump to what she was doing. She was taking 2 shapes, Q and the same shape in a different pose P, which is partial. So you just get only part of the shape. And she trained a network to use the PointNet as an encoder to decode a way of translating Q into the pose of P. Okay? So if Q is given as a whole and P is given as a part, what you do is you do this reconstruction where this was the actual ground truth. So this is using a network for this kind of reconstruction tasks. And again, the network is this PointNet used as a shrinking mechanism, as an encoder, and then another PointNet inverted, and you can take the surface Q and you can deform it so that it would stand as if it is P. Now you can see that there is a small point here that would tell the left hand to be somewhere there. What else did we do? With geometry processing, and this is an old story now, we used to scan about 400 kids in order to produce baby-fitted inhalation masks. This is a project with Israel Amirav from one of the hospitals in Israel. This is how a regular mask used to look like, and this is how the new, it's not new anymore, I think it's in the market for 5 years now, looks now, but this is using geometry processing in order to help the society. Yet another image. And another project together with Alon Wolf and our student Benjamin Greusser is to look at subjects who have scoliosis or problematic, some deformations in the back, and without projecting anything which is like X-ray on CT or something like that, be able by just 3D scanning, by just 2 eyes if you'd like, without any radiation, extract the kind of deformity that can be predicted. So this is an ongoing project. Benjamin is now probably would take it into some private company, but we have a really nice set of results that claim that you can take with a regular image the shape of a back and predict what is the internal deformation and actually provide a way of fixing this kind of deformation.